Mendel imagined that in the production of a sperm or an egg cell, the gene pair is split apart, with each gene ending up in a separate germ cell. Mendel called this process segregation. He also imagined that the direction of segregation is totally random. It can just as easily happen the other way around. So for any particular germ cell, which one of the gene pair it receives is totally up to chance, like the flip of a coin. This became Mendel's first law of genetics, the law of random segregation. This is how Mendel explained his results. His parent plants, the P generation, bred true for color. This meant they each had a pair of matching genes. The purple plant had 2 dominant genes, and the white one had 2 recessive genes. A genotype with 2 copies of the same gene is called homozygous. When the parent plants were cross-fertilized, they each contributed 1 flower color gene to the next generation. Since the purple parent had only dominant genes to contribute, and the white parent only recessives, each offspring had a genotype with 1 of each. This kind of mixed genotype is called heterozygous. A dominant gene trumps a recessive gene, so every plant in the F1 generation had purple flowers. The purple F1 plants were then allowed to self-fertilize to produce the next, or F2 generation. According to the law of random segregation, for any particular germ cell, the odds are even as to which gene it receives. So about half the sperm and egg cells carried the purple gene, and about half the white one. As a result, for any given fertilization event, there are 4 equally likely outcomes in terms of genotype. A purple pollen can fertilize a purple egg. A purple pollen can fertilize a white egg. A white pollen can fertilize a purple egg, and a white pollen can fertilize a white egg. The homozygous purple genotype produces a purple phenotype. The homozygous white genotype produces a white phenotype. Because the purple gene is dominant, both heterozygous genotypes produce purple phenotypes. What we are left with are 3 purple plants and 1 white one, the same 3-to-1 ratio that Mendel found in his various F2 generations. His experimental results were now explained.